Hello, my gentle, and of course very modern apes. Welcome to Gutsick. This is a place where I talk to you guys about cool science stuff, and I spend a whole lot of time debunking pseudoscience, and that pseudoscience that I spend the most amount of time debunking is creationism.
You gotta be able to take abuse, you gotta be able to have people talk crazy about you. In this business, you gotta be able to have people disrespecting you. And you gotta smile and you gotta pretend like that's okay.
The people who leave comments like why bother with creationism have been real quiet lately, for reasons that are probably not very difficult to discern, but it remains the case that debunking creationism is often extremely frustrating. It's insanely rewarding too, don't get me wrong. I get countless emails from people, religious and irreligious alike, who are giving me compliments that I don't deserve, and thanking me for helping them leave creationism themselves or giving them the resources that they need to combat creationists in their lives, whether it's personally or at school board meetings. But I think you have to understand that spending time debunking people who believe that they are divinely correct often feels a bit thankless. It is worse than Sisyphean. Truly, I can't even count the number of times that I have had a specific creationist argument given to me, spent hours debunking this argument, sometimes over multiple videos, sometimes across several months, occasionally going back and forth with specific creationists, only to, at the end of the day, having made my point, watch as they simply pivot with a brand new, completely unrelated argument that supposedly supports creationism, with no acknowledgement that the past argument was incorrect, no retraction so that their viewers or readers can understand that that argument was incorrect and know not to use it, and worst of all, sometimes, and the period of time depends on the creationist or creationist organization in question, that old argument comes back with the criticisms unaddressed. It is the same unchanged argument, pitched again, as if it is something novel.
And here we go. I hope you're hungry for nothing.
It drives me crazy. It's like if Sisyphus got to the top of the mountain, he's rolled his big boulder to the summit, only to turn and see a different mountain directly adjacent to him, and the flag has been placed there instead. He's frustrated, so he goes down to the bottom, rolls the boulder up that new hill, and when he gets to the top of that one, the flag is back on the original mountain, or on a third mountain, or one of millions, if not billions of mountains in the surrounding region, because this is not a single mountain, it is a mountain range. So why does Sisyphus do it? Why do I do it? Why do we continue at our task knowing ultimately it is futile? Do we do it because we're sick? Because we're masochists? No, we do it for the love of the game. One must imagine Sisyphus happy. Also, debunking creationism forces me to learn new things. I have to step out of my comfort zone and dip a toe into areas that I don't normally study. And that's fun and rewarding and cool and interesting. And it also provides resources to other people who might be asking the same questions. And every now and again, if you spend enough time at this, debunking creationism and taking their ideas seriously, an argument might actually break through. I want to tell you guys about how my work here on YouTube has helped kill a creationist argument. Probably for good. A video that I made severely criticizing the work of young Earth creationist and geneticist Jeffrey Tomkins caught the attention of a different young Earth creationist named Rob Carter.
She called Tomkins my brother in Christ. She called me that also. Eric, I don't know if that's sarcastic or if you're actually calling on the name of Jesus.
Who, in his investigation of my own work, came to agree with my conclusions that over a decade of Jeffrey Tomkins's work was simply incorrect.
And I agree with you on some of the most important parts, but there's a couple of things that I'm going to say in a little bit.
Because of this, the already waning frequency of creationists invoking the studies of Jeffrey Tomkins have decreased even further, to the degree that all of the most recent videos that I have seen covering the subject in question don't even mention his name. Meaning my work, triggered a series of events that led to the death of Jeffrey Tomkins' arguments, which, if you've been haunting around this area of discourse for long enough, you know was a pretty popular argument to invoke. That's for you short-form content aficionados out there. But for the rest of you who are no doubt wondering, what was the argument? What happened? How would a creationist end up agreeing with you, gutsickgibbon, to the point that the previous argument basically has just withered away and died? All phenomenal questions. I can't wait to answer every single one. But for those of you who have been around a bit longer, you already know what this is about. This tale concerns the human-chimpanzee genomic similarity and how creationists have slowly come around to a more conventional perspective. How similar are humans and chimpanzees? This is a question that's really important in the conversation of conventional science versus creationism because they make pretty distinct predictions. In the case of evolutionary theory, the idea is that if humans descend from more basal ape ancestors, then we are apes which descended from other apes, and that should reflect in our physical characteristics as well as in our genomes. If you are a creationist, you usually hold to some idea of special creation of humans, which originally reflected in creationists of all stripes insisting that humans are not apes and we are not related to any other apes, and in fact are very different from all other living things, but at minimum should always predict that there is a clear and identifiable biological distinction between humans and all other critters. Young Earth creationists believe that God created the Earth more or less in its present state 6,000 to 10,000 years ago, and that the entirety of the geologic column is due to a global flood that happened about 4,400 years ago. This means that all organisms are not related through common ancestry, but distinct groups exist which are unrelated from one another. Humans are one such distinct group. Now, the details of what falls into that created human group differs depending on the young Earth creationist or creationist generally that you're talking to, but virtually all of them maintain at least one similarity, which is that the human group, whatever falls into it, maintains a discontinuity with all other apes. So a young Earth creationist would say that 6,000 years ago, God creates all the different kinds of animals in these archetypal groups, and humans are one such group, and then the other apes are other groups, right? Either they are all created together as an ape group that is different from humans, or each ape genus is its own created kind, and these created kinds are unrelated to one another, right? So chimpanzees might not be related to gorillas from the perspective of a young Earth creationist, or all the apes might be related to one another, but none of them are related to humans, and none of them are related to monkeys, and so on and so forth. And there is a deep, deep history supporting these perspectives from a creationist and from a conventionally scientific perspective. As such, the question, how similar are humans to chimpanzees, or any ape for that matter, has a lot of bearing on these two models, if you will. So let's start with this. Why does conventional science argue humans are apes? We humans are apes. We are members of the superfamily Hominoidea and the family Hominidae. This means we are specifically great apes. The reason humans are classified this way is because we have all of the physical characteristics that designate our position with the other apes. Like them, we have internal tails, highly mobile shoulders, a Y5 molar pattern, dexterous hands and wrists, long gestations and long developmental periods, complex social systems, and large brains for our body sizes. We have all the same bones, muscles, and organs, and even our brains have all the same regions, with humans merely emphasizing portions related to cognition. This is why, although all great apes use tools, human tools are the most sophisticated. The difference between humans and other apes is one of degree, not kind. The fact that humans are so closely aligned with the other apes has actually been recognized for centuries, if not millennia. Linnaeus, the father of taxonomy and a creationist himself, even recognized this, choosing to elevate the other apes to our level rather than separate us out. And actually, since I created this clip, I've learned that it was actually Edward Tyson in 1680 that first made the case to the scientific community that humans are ridiculously similar to chimpanzees. He did this by dissecting chimpanzees and noticing the reverse, that they were ridiculously similar to humans, even when it came to the brain. Darwin fleshed this
idea out, of course, and after decades of intense scrutiny, there can be no denying that humans are physically apes just as dogs are physically canines. But even with that understanding, it was thought that humans were a very distinct ape indeed.
We thought ourselves special, apes, if only barely. Even before genetics came along, people anticipated being very similar to the other apes, but they still grouped all of the other apes in their own family, Pongidae, as compared to the human and sort of hominin family, Hominidae. Basically, among the great apes, humans were the odd man out, the most evolved, the most arrived, and gorillas, orangutans, and chimpanzees were all more closely related to one another than any of them was to humans. We were the standout. But genetics, specifically genomic sequencing, took that idea, took it behind the barn, and beat it to death with a rusty pipe.
The human genome was sequenced in 2002 and the chimpanzee genome 3 years later in 2005. This allowed comparisons to be made in full rather than the shorter sections of the genomes that were being compared before, and the results were striking. In coding base pairs, that is, the regions of the genome that code for proteins, humans and chimpanzees were 98.8% similar. When indels were included, so a full genome-to-genome comparison, that number hashed out to be 96%.
And worse yet for the human exceptionalists, as the early draft genomes came out for gorillas and for orangutans, it came to be known that humans and chimps were one another's closest living relatives, to the exclusion of gorillas and to the exclusion of orangutans. It wasn't just that chimps were our closest relative, we were theirs as well. Chimpanzees share more genomically with humans than they do with gorillas. So instead of the picture being like humans and then we have a group of cousins, which are the apes, really what it looked like was humans and chimpanzees as a pair of siblings, as sister genera. And then the cousin to the two of us was the genus gorilla, and then the second cousin to that triad was the orangutan. Tragically, the gibbons are the most removed amongst the apes, amongst the hominoids. You can't always get what you want. The fact that humans are 98.8% in coding base pairs and 96% in full genomic comparisons similar to chimpanzees is bad enough for creationism because this runs directly in line with evolutionary theory and directly contra to what creationists had been saying for decades. But I think really what knocks this out of the park and makes this a type 1 experiment, Rob Carter, is the fact that chimpanzees are also that similar to humans. They call us their closest living relatives. But we already have a little bit of a problem, don't we? Because from a genetics perspective, when we ask the question, how similar are humans and chimpanzees, the answer is, it depends on what you're comparing. If you're looking at those protein coding regions, it's 98.8%. But if you're looking at the full genome, it's 95 to 96%. And those aren't even the only ways that you can compare genomes. For instance, researchers like Richard Buggs and content creators like Minute Earth have pointed out that the alignment percentages that we currently accept take into account the fact that humans and chimpanzees have differently sized genomes, with humans having approximately 3.2 billion base pairs and chimpanzees having approximately 3.8 billion base pairs. So how do you compare those? Like, let's say, for example, we were comparing two Bibles, and in one copy of the Bible, the book of Deuteronomy was cut. It was just absent. There was a printing error. And in the other copy of the Bible, the book of Genesis is in there 3 times due to another printing error that accidentally duplicated it. Well, how similar are those two books? The Bible that doesn't have Deuteronomy in it can fit and be found in its entirety in the Bible with 3 copies of Genesis. But the Bible with 3 copies of Genesis cannot be found in its entirety in the Bible without Deuteronomy because it has 3 copies of Genesis. It's significantly larger. But the differences are due to duplications of a book that is found in the Bible without Deuteronomy. What if there was an additional printing error that caused the word Lord to be deleted just from 2 of those 3 copies of Genesis in our triple Genesis Bible? How would you compare it then? If you tried to line them up side by side, you would run into a ton of alignment errors because even though the vast majority of the words are the same, they don't align one to one because of the absence of words or the addition of words. In addition to that, there are also portions of both the human genome and the chimpanzee genome, in every organism's genome actually, that are incredibly repetitive. So you might have like ATA, ATA, ATA over and over and over again. So how do you compare those regions of the genome? Sometimes there are inversions where an entire sequence will be inverted or will be in the wrong way, and that can occur with a single mutation, or enormous pieces of the genome will be deleted or placed in the wrong spot or duplicated. Now, thankfully, comparative genetics work can take these kinds of things into account, and it's actually how we get virtually all of the percent similarity numbers between dog breeds, between people, between humans and chimps. But what happens if you just ignore the sources of those differences? If you don't pay any attention to the types of differences and you just line the two genomes up side by side and brutally compare them nucleotide by nucleotide? How similar would they be then? Geneticist Richard Buggs has done the math on this, and if you do it his way and absolutely maximize the differences between humans and chimpanzees, you get a similarity of about 84%. Now, creationists cheer at the thought of that, but there is a massive problem with this type of calculation from their perspective, which is that if you go nucleotide by nucleotide and aim to maximize the differences between two organisms in their percent similarity, every single pair of organisms are going to widen by that method, including humans compared to other humans. Typically, by the means that we get 98.8% for humans and chimpanzees, humans are about 99.9% similar to one another. But if you use Buggs's method of maximizing differences and not taking into account the type of differences at play, humans will drop to only being about 94% similar to one another. But the craziest part is that it doesn't matter how you compare genomes. If you do protein coding regions only, full genomes taking into account the types of differences, or Buggs's method where you go nucleotide by nucleotide, humans and chimpanzees will still be one another's closest living relatives. We will still be most similar to one another than either of us is to a gorilla or to an orangutan, and will also be more similar to one another than lions are to domestic house cats or rats are to mice. And that's a critical part about this, having a comparative pair of organisms to ground the human and chimp genomic similarity. So one thing that everybody seems to agree on is that genomic similarity reflects relatedness within people and within species in general, right? So you share more of your genome with your family members than you do with a stranger. You also share more in common genomically with your cousin than you do with a stranger, but you don't share as much with your cousin as you do with, say, your parents. This kind of thing works so well that it holds up in court via a paternity test. We all just recognize that the more of your genome you share with someone, the more closely related you are to them, and the less of your genome you share with someone, the less closely related you are to them. You can also do this within breeds of animals. So you can compare all of the different dog breeds to see when specific dog breeds emerged and which dog breeds are more closely related to others. And you can do it at the species level, so you can see which species are more closely related to one another. This is something that creationists will accept to a degree. For example, they are more than okay with looking at the similarity between cats and lions and saying that they're so genomically similar that they are actually related to one another. They descend from the same created kind in the Garden of Eden. From an evolutionary perspective, everything is related to everything else, and we all have common ancestry up to a last universal common ancestor, which emerged sometime between 3.8 and 4.1 billion years ago. So we have a nice Type 1 experiment here. A Type 1 experiment is an experiment whose results can differentiate between two hypotheses. It can allow you to accept one and sort of preclude the other, or at least do away with it for the time being. Because if overall genomic similarity does indicate relatedness, at least within created kinds, then you can compare the genomes of organisms which creationists accept as related and contrast that percent similarity with a pair of organisms that creationists insist cannot be related. Evolutionary theory would predict that the percent similarity should fall in a nested hierarchy that should more or less reflect hierarchies established by morphology and the fossil record. It can thus be denied if there are large discontinuities that are discovered in both functional and non-functional regions of the genome between organisms which ostensibly evolution would predict as closely related. Denying creationism as a hypothesis is a much simpler endeavor. All that needs to happen is the similarity between two organisms that creationists insist are not related, like humans and chimpanzees, should be higher than the percent similarity between any pair of organisms that creationists accept as related, like cats and lynx, or rats and mice. And critically, this pattern should hold no matter how you are carrying out your comparative genomics. It should hold for the protein coding regions, it should hold for the entire genome, and it should hold for brute, raw, nucleotide by nucleotide comparisons as well. Put a pin in that for now. Let's just appreciate that conventional science says that humans and chimpanzees, as well as bonobos, are 98.8% with regard to coding base pairs, and they are 95 to 96% when looking at the full genome, and that is, as anybody would intuitively think, a lot. Too much if you are a creationist. Enter Jeffrey Tomkins. Jeffrey Tomkins is a young Earth creationist geneticist, and he found that high human-chimpanzee genomic similarity dubious, or as I suspect, inconvenient. So he has spent the past several years publishing study after study after study in the Creationist Journal, the Answers Research Journal, which has no secular peer review, arguing that the number actually isn't that high. It's not as high as they might have you believe. In fact, it is significantly lower when you do it right.
Genome-wide DNA alignment similarity for 40,000 chimpanzee DNA sequences queried against the human genome is 86 to 89%. How genomes are sequenced and why it matters, implications for studies in comparative genomics of humans and chimpanzees. Comparison of the transcribed genomic monkey regions to the estimates of nearly identical human chimp DNA similarity, evaluating comparison analysis, trace analysis, chimpanzee context to genome, wide DNA average blast, and identities at 84%.
You're under arrest, Nimrod.
Now, the actual percent similarity that Jeffrey Tomkins has come to as like the true similarity for humans and chimpanzees has ranged from 70% to about 89%, which even at its highest percent is significantly lower than the conventional numbers, but it typically ranges in his most recent sets of statistics from 80 to 89% for an average of, let's call it 85%.
I found out that the human and chimpanzee genomes were no more than 85% similar.
Now, conventional science took virtually no notice of this for many reasons, but the simplest of which is one guy saying that he found results that contradict what hundreds, if not thousands of other geneticists are getting is not cause for everyone to drop everything and acquiesce to him. And in fact, the results continued to come out in favor of conventional science as more great ape genomes were sequenced in more depth, and eventually all the way up until the telomere to telomere full complete human genome, which was only completed in the past year or two. But to creationists, especially young Earth creationists, this was a huge deal. And Jeffrey Tomkins' numbers started popping up everywhere, even in organizations he is not explicitly attached to. Creationist watchdogs like myself immediately thought something was off, and many folks posted their critiques of Jeffrey Tomkins' work on places like the Peaceful Science Forum or the Debate Evolution subreddit. The one who probably did the most work was Glenn Williamson, or Ruhif. Glenn is an absolute monster when it comes to computer programming, and he has kept up with Jeffrey Tomkins' work very consistently. He still keeps up with it to this day and offers criticisms from a perspective of computer programming and statistics. It was actually Glenn that found out that Tomkins' originally published number, that humans and chips are only 70% similar, was due to a bug in the version of BLAST that Tomkins was using. But like I said, Glenn has meticulously combed through all of Jeff Tomkins' papers and demonstrated why they are incorrect using the best possible methodology. trying to replicate them himself. And it was Glenn's work specifically that inspired me to run the tests that I ran. I grew curious. If Jeffrey Tomkins is getting only about 85% similarity between humans and chimpanzees using this very specific set of methods carried out in a program called BLAST, what kind of results would he get for other pairs of organisms? I wanted to run that type 1 experiment myself. Because the conventional numbers are already in. Humans and chimps in protein coding regions are roughly 99% similar to one another, and that number drops to about 95% when you're comparing lions and tigers, or African and Asian elephants. It drops to about 85% when you're comparing rats and mice. And remember, these are protein coding regions. This is like the maximum amount of similarity. Now, I had a hunch going into this that the reason Jeffrey Tomkins was getting that around 85% number was because he was just doing what Richard Buggs had done. He was attempting to maximize the differences between humans and chimps by counting every nucleotide that was different in a direct alignment as different, as going towards that percent difference, regardless of the context of the mutation or where it was found in the genome or the sizes of the genomes. That's what I thought was going on. And likewise, I predicted along those lines that we would expect that all comparisons between organisms should broaden using that criteria. I was planning on running these experiments and then coming back and doing a video on it. And if things matched my hypotheses, I was going to say, yes, if you use Tomkins methods, you can get humans and chimps to be like 85% similar to one another, but humans and humans will drop to around 94% similar, much more different than under conventional means, and other pairs of organisms will drop even further down than the human and chimp number. So lions and tigers will go from being 95% to somewhere in the like 70% range, and rats and mice will go from being 85% to somewhere in the 60s or even the 50% range. That was my expectation. And it was going to be very satisfying because I was going to be like, see, Tomkins, humans and chimps are still one another's closest living relatives. This doesn't actually help you, and you still can't discover this barrier, genetically speaking, for where the created kinds are. That is not what happened, though. I published my results here on YouTube, and the video did pretty well. Well enough to get the attention of a colleague of Jeffrey Tomkins's, Robert Carter. My video was pretty inflammatory, so it's understandable why it caught Rob Carter's attention, but the reason it was so inflammatory is because over the course of running my tests, a process that took several months and a whole lot of effort, I came to the conclusion that Jeffrey Tomkins is either grossly incompetent at what he does or a bold-faced liar. I still think that. Because you see, it turned out that Tomkins didn't get that 85% number because he was doing what Richard Buggs had done, not even close. He got that number through an increasing cascade of poor genetics work that he documented paper by paper over the course of like a decade. It was mistake after mistake after mistake, and the mistakes stopped looking like mistakes to me, and they started looking deceptive when I realized they appeared, at least to me, to be made in such a way so as to corroborate one another. To briefly summarize, Tomkins's methods can be split into two sets of criteria. The criteria that he used pre-2015 and the criteria that he used post-2015. And these two sets of criteria, basically the parameters that he used within that genome comparison program BLAST, were very different. And I found that odd. Still, using those criteria, I compared humans and chimpanzees as well as a bunch of other pairs of organisms, and what I found was that it doesn't matter which methodology you're using, humans and chimps always end up being more similar than many other pairs of animals that I know Tomkins would accept as related. And in fact, for his pre-2015 methodology, there was something grossly wrong with how he was running his analyses because I couldn't get a 100% similarity of a sequence of human DNA to itself using his methods. The newer stuff, that post-2015 methodology, was just boldly wrong because Tomkins doesn't weight any of his sequences. So he basically has all of these different little chunks of human and chimpanzee DNA that he's comparing to one another, and he is getting an average. He's taking an average of all of these similarities, but the sequences are not all the same length. So he is weighting a 300 base pair long sequence, for example, that is only 50% similar between humans and chimps as equal to like a 200,000 base pair sequence that is 99% similar between a human and a chimp, which is, of course, erroneous. If you're at all interested in this subject, please watch my video on it because I go piece by piece, step by step, and explain why all of this happened in a very, again, suspicious manner, in my opinion. It's not just that Tomkins is wrong, he's wrong and highly suspect. And that video is also good because I go over some interviews that Tomkins gives, and it really seems to me like he either doesn't know what he's talking about or is deliberately lying and is maintaining this interview to interview. The tea is hot. Please check it out. That's one of my favorite videos that I've made, and I don't think that it has enough views. Like I said, I still think that Jeffrey Tomkins is either incompetent or dishonest. My current opinion is that he's probably like a healthy mix of the two, but I came in real hot with these accusations. Again, they were all my opinion, but I did voice that opinion, and I think I came with receipts for that, which is why it caught the attention of one of Tomkins's colleagues. Rob Carter responded to my accusations with a little video that really pissed a lot of people who are fans of me off, because in it, he essentially says that I made some mistakes in my analysis, but then he was like, I'm not going to get into those.
She's trying to analyze his work in genetics, and she's making some subtle mistakes of her own.
Now, you know, I'm only human, so of course I make mistakes. I make mistakes all the time. But my question is, like what?
Which I'm not going to go into, but it's clear that she's not a geneticist.
That's right, baby!
As Robert Carter puts it, he was gang tackled for this.
I also expected to be gang tackled.
Obligatory, Rob Carter is one of my favorite young earth creationists, and one of the reasons for that is that he films his videos for his own channel, Biblical Genetics, like on site at various locations across the United States. So it's like, where in the world is Rob Carter this time? And I think that that is fun and very charming. But because of the criticisms that he got, he decided to undertake a larger analysis to see if I was correct, I guess, and to look a little bit more deeply into the claims of Jeffrey Tomkins as well. So Dr. Carter proceeds to take countless hours learning how BLAST works so that he can download genomes and try to recapitulate my methodology as well as Tomkins's methodology to see exactly what's going on, if my accusations were appropriate, if Jeff's conclusions were appropriate. And the conclusion that he came to is that, one, you shouldn't be using BLAST for this analysis.
Don't think that, first of all, BLAST is an appropriate tool to be using.
2, he couldn't recapitulate my work.
I can't validate her work.
3, he couldn't recapitulate Tomkins's work.
I can validate Tomkins' results. Now, I don't agree with his conclusions, but if I do what he did, I'll get the same numbers, essentially, in my small data set. Okay, I'm adding a second note in post-production here. I did a lot more work on that question of, can I reproduce Tomkins' numbers? Turns out that I can't.
And 4, and most importantly for my entire role in this, Jeffrey Tomkins was wrong.
And I agree with you on some of the most important parts.
Not some of his numbers were wrong, all of his numbers were wrong. Now, I had made some mistakes in my analysis. My explanation for gapped versus ungapped was incorrect, and I also incorrectly applied Tomkins's methodology to the wrong version of BLAST. So my response to Carter's series of videos was to rerun everything the right way. And my conclusions held completely, down to the part where Tomkins's methodology incorrectly returns a non-100% similarity of a chunk of the human genome to itself. It just wasn't in BLAST, it was in 2 other programs called NUCMER and LAST-Z. Again, please watch that video if you want to see those results in depth, but using Tomkins's pre-2015 methodology, humans and chimpanzees were more similar to one another at around 90% than dogs were to maned wolves, than dogs were to foxes, than lions were to lynx, than water buffalo were to cattle, than macaques were to baboons, and of course, than rats were to mice. Tomkins's biggest issues were the fact that he just didn't weight his sequences, a problem that plagued all his methodologies, and rendered his results worthless noise. Carter's conclusion for how similar are humans and chimpanzees was, we can't really know yet. Like, a lot of work needs to be done on the creationist side, meeting all of their sort of appropriate assumptions, I suppose, to come to a number that they're happy with. but he was very vocal that it was not going to be 84% or 85%. It was going to be much closer to the conventional number.
Because I don't think we're going to end up with Tomkins' 85% similarity value. It's going to be higher than that, but it's not going to be for the reasons why some of the evolutionists are saying.
He never officially responded to my triplet of videos, which I understand because it was a lot of content, and I also understand because Carter and Jeff are like friends. They like know each other and are buddies, I think. So it can't have been easy for him to come out and basically say that his colleague has been wrong the entire time, agreeing with like the evolutionists who have been saying that for many years. I actually reached out to Carter via email at one point, and I was like, are you gonna go back to all of the times that you've endorsed Jeffrey Tomkins' work and this specific number and like put a disclaimer on it? The reason I was asking was because if the answer was no, I was planning on putting a spotlight on all of those times in a video on my channel. This was going to be a resentful jab at Carter for finding out that he was wrong, but not going back and correcting himself for future reference. It was really tough for me in my video to admit that I was wrong about my initial methods, to eat crow and realize that I'd made some mistakes in my methodology. And even though I fixed it, and even though my conclusions held, there was a part of me that was kind of irritated at Rob Carter for pointing it out when his own work was showing that he was also wrong, except instead of it being one video a year ago, it was multiple blog articles and videos across several years. But I didn't do it because one, that's really petty and kind of shitty, and two, because Carter was the only one who bothered to check. He was the only young Earth creationist who looked at Jeff Tomkins' work and said, are we sure about that? And peer-reviewed it. Certainly other creationists have looked at Tomkins' work, like Todd Wood, for example, but Carter wanted to try to recreate it himself. And in that way, like I said, he peer-reviewed him. Now, granted, it was through me, but Jeff Tomkins is his colleague, and Carter found out that he was wrong, and very overtly so, and then Rob was honest about it. That's more than most young Earth creationists can say, and it's certainly more than Jeffrey Tomkins can say, who is still obstinate that he is correct in these papers, at least so far as I can tell.
I have no idea what this Gutsick Gibbon girl did, but yeah, you're right. They kind of feed off of each other.
Okay. Let's kill him. Let's kill this guy. Let's beat him to death with hammers.
So I had other things to worry about, and I was happy with where we stood. Everyone agreed Jeffrey Tomkins was wrong, and I let it go. I figured Rob Carter was going to publish his write-up in the Creation Journal about the revised similarity between humans and chimpanzees, which I knew he was working on, and that was going to be the end of it. And it seemed like it was the end of it, because he was publishing videos on his regular YouTube channel that didn't really have all that much to do with the human-chimpanzee genomic similarity conversation. So imagine my surprise when a trailer showed up on my YouTube feed.
Hello friends, I am Dr. Robert Carter. As a member of Logos Research Associates, I have been asked to bring to you the latest information on human-chimpanzee similarity or dissimilarity.
Humana, what? I wasn't expecting this. Interesting, Rob. Tell me more.
I'm going to be using the most modern genomes available. These are the first telomere to telomere genomic sequences. So any old ideas we had about the similarity of humans and chimpanzees is going to be contradicted because the data are brand new.
Oh, wow. Interesting. Very, very interesting way of framing this. If you've been keeping up with this and if you've been paying attention to the story so far, you're probably thinking the same thing I am, which is this is a very diplomatic way to frame things if you're friends with Jeffrey Tomkins. It isn't that Jeff made gargantuan, conspicuous, and ubiquitous mistakes across a decade of research. No, it's just that the data that we have now is the newest so far. And, you can't blame him for not having access to the newest data so far. It didn't exist yet.
That's not your fault. It's easy to get fooled when you're close to somebody like that. However, on our end, he looked extremely untrustworthy.
We did not have this information until just the last year or so.
I see you, Rob. I get it. but I see you. Dr. Carter then goes on to explain some of the tests that he's going to be talking about at this Logos Research Association interview, and some of it had my jaw on the floor.
So I'm going to be aligning human chimpanzee chromosomes and showing you the results. I'm going to be comparing human chromosome 2 to the 2 smaller chimpanzee chromosomes that do actually have a very high match.
Why would she post that?
That's like Jeffrey Tomkins' other big thing. Debunking the human chromosome 2 fusion is the other big thing that he spent an insane amount of time on over the past decade. It's human-chimp chromosomal similarity and trying to prove that human chromosome 2 is not the result of an ancestral fusion event between ancestral ape chromosomes 12 and 13, or what we call in chimpanzees 2A and 2B. Rob!
I'm going to look at what's called synteny, that's gene order along the chromosomes. And we're going to be tackling some of the older creationist ideas and some of the older evolutionary ideas and basically correcting the record a lot.
Correcting the record a lot. I'm seated. I'm sat. Now, to be clear, there are other young Earth creationists that accept that a fusion event occurred in the human lineage. There are even young Earth creationists that accept this idea, that a fusion event occurred, like, I don't know, during Noah's Ark or something like that, and that's why human chromosome 2 looks like a fusion of 2 other chromosomes. More on this later. But, like, specifically couching this in a conversation that's already about debunking some of like Jeffrey Tomkins' other work. It's like I'm going to be doing these 2 things, and those 2 things are specifically the 2 things that Tomkins made the biggest case for and kicked up the most amount of dirt for and, dedicated a decade of his life to.
Sadly, I'm going to have to contradict some of the most important papers so far written by our side. Because the information is the information, data or data, and you have to take it at face value, and you have to say, okay, this is what we have, now we can have a conversation.
What? Who is this? Are we talking to Rob Carter's innie? Is he severed? Yeah, that's right, I'm up to date on my pop culture. Okay, in all seriousness, massive W. Thank you, Robert Carter. Thank you, Creation Ministries International. I could not appreciate this more. Yes, in fact, the data are the data, and I will take a moment to say here that it's true. A high percent similarity between humans and chimpanzees and an evident fusion between ancestral chromosomes that appear to be highly similar to chimpanzee chromosomes 12 and 13 do not necessarily mean that you can't be a young Earth creationist. Now, in my opinion, they are strong pieces of evidence that run contra to young Earth creationism, particularly because they were predicted ahead of time. But I would much rather see young Earth creationists attempt to accommodate correct data instead of deny or ignore it. But as amazing as all of that is, it is true that Dr. Carter himself is responsible for pushing those Tomkins numbers a lot on his blogs and in videos and so on and so forth. I mean, I guess it would be way too much to ask to get a correction on that.
I'm actually going to have to contradict myself a lot because some of the things I've written, some of the ideas I've supported, some of my initial analyses turn out to be not right.
If I'm dreaming, let me sleep. Don't wake me up. Do not let that alarm sound. This is more direct engagement with this specific data from a non-new creation young Earth creationist that I have seen in a very, very, very long time. Let me sleep. Let me have this. I mean, what the hell? That is so cool. I know how much that hurts. I just did this on this channel. I mean, I guess it was like a year and a half ago, but still, it hurts so badly to admit that you were wrong and to do it on a camera in front of a lot of people. That is brave and awesome. With due credit out of the way, where is this going?
So as we go through this, we're learning, but we're also going to be seeing the differences between humans and chimpanzees, and they are, in fact, still quite profound.
Interesting. I would expect them to be quite profound because humans and chimpanzees are different species and we diverged from one another like around 7 million years ago. I would expect the differences to be present and I would expect them to be profound as they would be for any sister genera of that nature. But I think that's probably not what he's getting at here.
Humans and chimpanzees are going to end up being probably above 90% similar. But we're also going to see that there are massive stretches of extremely similar DNA. Like 98, 99% similar for 100,000, 200, 300,000 bases in a row. But those are separated by completely non-comparable DNA.
There are quite a few numbers above 90%. I wonder if he thinks it's like 91, or if he thinks that it's like 95 or 96%. Does it look like the conventional numbers or not? Now, as for that second half, right, I expect that there should be enormous stretches of extremely similar DNA separated by decent-sized differences. Like, again, humans and chimpanzees are different species, and we have large behavioral differences. That is not shocking to me. Humans aren't chimps, and chimps aren't humans. We're just one another's closest living relatives.
So the question is not going to be, how similar are humans and chimpanzees? That's actually a question that neither side predicted initially.
I mean, no one put a number to it. Like, it wasn't like we're in 1963 and young Earth creationists are saying that humans and chimps are only 50% similar, whereas conventional scientists and geneticists were saying, actually, it's going to be 99%. That wasn't happening. I mean, the genetics work for chimpanzees wouldn't have been until like the 1970s, so I guess 1963 was a bad year to pick, but the point still stands. The earliest work on genetics that included chimpanzees and was comparing humans to other apes, like, people weren't thinking that this was going to be 50% similarity. We had a lot of very basic genetic studies already underway comparing humans and chimpanzees, and that was predicated on prior immunological work that showed that humans were very similar to the other apes. So it's true that conventional science said that they were going to be very similar, and creationists said that they weren't going to be similar at all. Remember, they said that humans aren't even apes. So I think it's wrong to say that the high similarity between humans and chimpanzees, and specifically the high percent similarity between chimpanzees as compared to humans, right, so we're one another's closest living relative, actually really gives a point in favor of conventional science as compared to young Earth creationism. This number is not agnostic. It does have bearing on the conversation, especially when compared to other groups of organisms that creationists would accept are related, like the genomic similarity between rats and mice.
And it doesn't matter either way to either side, because if we're 80% similar, the evolutionists would just back up the clock. If we're 99.99% similar, the creationists would just say, well, that's how God made us.
Conventional scientists would investigate why the percent similarity is so low. They would investigate the genetic processes at play in hominids to see if there was an increase in the mutation rate that might explain such a fast divergence. Yes, they wouldn't immediately be like, well, I guess evolution isn't true anymore. They would investigate it at great length, but let's not compare this to creationists putting their heads up and saying, oh, well, it's common design. Common design does not explain why humans are chimpanzees' closest living relatives, instead of chimpanzees being closest to, say, gorillas. It also doesn't explain why that similarity holds in both functional and non-functional regions of the genome.
So that's not the argument. The argument is, one, how many mutations? Okay, we now count those. Two, how long would it have taken? 3, where does the source of the new information come from? Because there are hundreds of genes, entire gene families that humans have that chimpanzees don't.
Okay, so the conversation is moving to a waiting time problem and like irreducible complexity kind of argument. Okay, we can do that.
That's the kind of questions that we have to ask. Both sides are predicting similarity. Both sides. Now, that number was never predicted by anybody, so we're going to be talking about that.
As I said, that's not right. The positions of creationists and conventional science could not have been more diametrically opposed with regard to their expectations for the genetic similarity of humans to other apes. It's true, no one put a number on it, but both sides were not predicting similarity. One side predicted similarity, and the other one said the polar opposite, to the degree that humans aren't related or similar at all to other apes.
So come for the show. The comments should be lively. Discussion afterwards might be a little sharp, but we'll see.
I am this close to changing the entire trajectory of my life and career so that I can devote my time to studying quantum physics, invent a time machine, and go back so that I can be there in the comments section for this conversation, because it already happened and I missed it. Rob talks a little bit about how all this information is already out there. It's not like this is the premiere of his position on this, because again, he did do that write-up for CMI, right, where he talks about how we're revising the human-chimpanzee genomic similarity, and I think that that's probably intentional. He doesn't want this to come across as like a rug pull on Jeffrey Tomkins, and he especially doesn't want other creationists to think that that's what he's attempting to do. Sure, I respect it. That's fine. I'm not trying to paint this as a rug pull. It's not like no one saw it coming. The signs were there that Rob Carter was going to be overturning the Jeffrey Tomkins numbers, thanks to me. I don't mean thanks to me like my work did it. I mean thanks to me like if I hadn't made my video, Carter wouldn't have responded to it, which means I wouldn't have responded to it, which means he wouldn't have done a deep dive on my work and Jeffrey Tomkins' work and then come to agree with me and virtually everybody else that Jeffrey Tomkins is wrong. So in that way, thanks to me.
I do this, though, as a service to the community. This is not, unknown to the world. I have an article in the Journal of Creation on this topic a year ago, and I prepared another one for submission for some upcoming edition. So this is information that the creationist world is talking about. We now know because we now have full genomes. So I hope you enjoy this.
Rob, what was that at the end? What did you just do?
Because we now have full genomes.
What in the eyebrow? Rob, either that is a covert signal that Robert Carter knows that new genomes are a poor excuse for covering up Jeffrey Tomkins's either incompetent genetics work or deceitful lies with regard to the human-chimpanzee genomic similarity, since he did it for over a decade. And Rob Carter knows that this is the case, but doesn't want to throw Jeffrey Tomkins under the bus, or he's just being a little goofy. That is on brand for him. Carter is a silly guy. Obviously, this little trailer fascinated me to no end, and so I went to go watch the full interview, the full presentation. It was 2 months ago. I have thoughts that I would like to share with you, and then I have a couple of thoughts with regard to the state of young Earth creationism and creationism generally on the human-chimpanzee genomic similarity and where it's kind of moved. You already know. I already spoiled it at the beginning. One thing you need to know about this presentation is that it is full of tentative disclaimers, where the host is telling us that part of what makes Logos Research Associates so important is that it allows creationists to kind of challenge one another and stay on the cutting edge of science.
The goal of our live stream broadcast is to provide you with the opportunity to hear from top researchers who will be conveying cutting-edge research for the Genesis creation account.
As a side note, cutting edge for creationism, absolutely. But cutting edge for science, no, not even close. If it was cutting edge for science, Carter would be publishing it in Nature or Cell or any legitimate genetics journal with extensive peer review. But that's not what he did. He published it in the creationism journals. That's not to debase the work that he's doing. It's just to say, cutting edge has levels to it. But you can really hear how tentative the host is being about the talk that Rob's going to give. And maybe that was at the behest of Dr. Carter himself, because Carter introduces himself. He's appreciative of the compliments that the host has given him. And then he begins his talk like this.
This is something that I've been both looking forward to sharing and dreading at the same time.
Right, we just heard those reasons in the trailer for this talk. Obviously, it's not fun to have to come out in your community and say that a colleague of yours was consistently and ubiquitously wrong over the course of a decade on several different topics. And it's also not fun when you're doing that and saying they weren't just wrong, but the people that were critiquing him were right. Not just on the subject matter, but in their critiques of your colleague.
I am going to get my button correct there. There we go. I'm going to be discussing my latest research on human and chimpanzee genetic similarity and dissimilarity. One of the reasons why I'm dreading this is, and not so much for the embarrassment, I am going to contradict some of the things that I've said before and some of the things that I've written before, but I'm a big boy. I can handle that. I understand that.
Yeah, but it still hurts and it still feels embarrassing, right? Like, I'm a tough person as well, but that doesn't make it any easier to get on camera and own up to a mistake that you made, even though everybody makes them, even though every scientist makes them, and even though making them and owning up to them allows us to move forward and is kind of a critical part of the process of investigating the natural world. And I think because of that, not everybody does admit when they've made mistakes.
Anytime you publish or say anything as a scientist, you're opening yourself up for contradiction. That's how science works. We advance by showing people in the past were wrong. And so that's just a fact of everything. And I'm not embarrassed about it. It's just a little humbling sometimes.
I mean, I'm embarrassed and humbled when I make mistakes. And I make mistakes all the time, right? I'm a scientist who talks about science stuff on YouTube, where tens of thousands, if not hundreds of thousands of people like fact check me in real time. Like, you're going to make mistakes doing that. And sometimes they're little and sometimes they're big. But one thing that my audience can always rest assured knowing is that when I'm called out on a mistake, I own it. I own it publicly. I correct myself, and then I do my best not to make that same mistake again. I like to think that that affords my content a level of transparency and a level of trust that not every channel or outlet for science communication out there has. I actually think it's a pretty good honesty litmus test. If someone has publicly made a mistake and refuses to acknowledge it, no matter how many times they're shown that they're incorrect, I think that that's pretty good support for you to like not trust that person moving forward.
What I'm afraid of, though, is that I'm going to have to contradict some of my fellows, and I don't enjoy that at all. But again, as scientists, I've spoken to all the people involved. They know where I'm coming from. There's nothing to be surprising here. But the ball advances down the field sometimes, and sometimes we have to let go of older ideas.
Right, so correcting your friends and colleagues sucks, but to me, that little anecdote is yet another reason why Jeffrey Tomkins is so shady, because Rob Carter, at least to me, this is speculation, what this little anecdote makes me think is that Carter reached out to Tomkins to explain kindly as a brother and as a friend why he's incorrect, and Tomkins is still just like, no, it's the children that are wrong.
Something else I'm concerned about is that I know that some evolutionists are tracking what we're saying on these subjects.
Yeah, that's true. I do track what you guys are saying on this subject. He's on to us. Scatter!
And they might misunderstand some of the things I'm saying, or saying, oh, look at that, they agree with us, evolution is true. No, that's not true at all.
This is how I know that this is in part like directly at me, because my thumbnail and title for the previous video that I made on Rob Carter and this entire incident was titled like, Creationist agrees with me, Tomkins wrong. But I think that it's also kind of telling to hear Rob say this, because the title says it all. I didn't say creationists agree with me, evolution right. I said creationist agrees with me, Tomkins wrong. That was the entire point of my experiments. That was the entire point of making the video on it, right? Like, I wanted to show that Jeffrey Tomkins's methods and thus results, and thus every creationist that I've ever heard parroting him, were incorrect to do so. Now, I do think that that has bearing on evolutionary theory being superior to every version of creationism that we have here in the West, but that wasn't the point. The point was to investigate one methodology by one young Earth creationist and explain why it's incorrect and why humans and chimpanzees are as similar as conventional science has been saying for many, many years. Anybody out there who's saying stuff like, oh, Gutsick-Gibbon made Rob Carter admit evolution is true, like, that's just blatantly false. And it's also conceptually false, because there are many young Earth creationists out there who do accept the high percent similarity between humans and other apes. They rationalize it away, but they do exist. And Rob Carter is moving towards being one of those types of young Earth creationists, I think. What he says next certainly suggests it.
But I am going to bring the similarity arguments a little closer to what the evolutionists have been saying for a long time, and a little farther away from what the creationists have been saying, myself included, for a long time.
Now, I have no idea how close to the conventional numbers he's thinking here. We know that it's above 90%, but is it like 91%? Is it 96%? I said this earlier, but it's unclear. If I had to guess, I would say that Rob is going to come out at the end of this and be like, humans and chimpanzees by my methods are like somewhere between 94 and 95% similar to one another. And he's going to take that as win because it's a reduction from 96% for the full genome. But as I hope you understand from the previous entire video that you've just consumed, it's not. It wouldn't even be a win if it were Jeffrey Tomkins' numbers specifically, because this number needs to be grounded in other pairs of comparative organisms. Now, that brute similarity is important to the conversation, and if it ended up being like 85%, I do think that that would be surprising to the community of biological anthropologists and paleogeneticists and things of that nature, because it would be such a big drop from what we had previously thought that it was, and it would imply a much faster mutation rate in hominins, for example. So the high similarity is important because it was predicted ahead of time by evolutionary theory, but it's not as important for this creation-evolution discussion as the comparative pair of organisms that grounds what that number actually means, right? Because again, and I cannot stress this enough times, I will beat this horse to death and then I will pound its carcass into a fine paste. Whether humans and chimps are 98.8% similar for the coding base pairs, 96% similar for the full genome, or 85% on nucleotide by nucleotide, that's always going to be a higher percent similarity than lions versus tigers, or African versus Asian elephants, or rats versus mice, all of which creationists accept as related organisms through common descent. So Dr. Carter goes into his intro, where he gives us his background as a marine biologist, and specifically the type of marine biology that he did was coral reef ecology, and he did a lot of genetics work within that, so he is an appropriate person to be looking into the genetics on this stuff.
Genetics is a passion of mine. I just want to know more about DNA and genes and how things are passed on.
I actually kind of love Carter's intro because he talks about what he did for his dissertation work, which involved taking the genes out of fluorescent corals and then putting them into fish. Like, I think they're like little tetras, but I'm not sure, which is just really cool. You can see it, the picture of his little fluorescent tetras that he has on his slide. I think that's awesome. I want more of Rob Carter doing conventional science, please. Before getting into the human-chimpanzee genomic similarity, Carter talks about the 3 types of experiments. Type 1 being an experiment whose results can differentiate between 2 hypotheses, supporting one and rejecting the other. Type 2, which supports one hypothesis but doesn't really say anything about the other. And Type 3, which doesn't really say anything about anything. And the reason he says this is so that he can assign the human-chimpanzee genomic similarity to one of these types.
I say that because the human-chimpanzee argument is a type 2 experiment. It doesn't matter to the creationist.
So I've said this already, but I don't think that that's true at all. I think that that only holds if you are working with an extremely modern, extremely liberal version of young Earth creationism. Human evolution, the idea that humans are apes who have descended from other apes, has always been the biggest bugaboo for creationism from Darwin's time to now. It was the last thing that people who came around to accepting evolution came around to accepting. And to this day, it is still a sticking point when polls are taken about public acceptance of evolution. Other animals are allowed to have evolved, but not humans, because we are special. As such, creationists, from the diluvialists of the 1800s to the fundamentalists of the 1920s to the evangelicals of the 1960s to the intelligent design advocates of the late 90s and 2000s, have really dug their feet in on this idea of humans being apes, something that the similarity of humans to chimpanzees and of that dyad to all other primates certainly has a huge bearing on. The high similarity absolutely has bearing on the conversation because it reduces the distance that humans have typically put between ourselves and the rest of the animal kingdom. But it also has bearing on the conversation because relatively the similarity between humans and chimps is higher than the similarity between chimpanzees and gorillas, or between chimpanzees and orangutans, or between humans and gorillas, or between humans and orangutans. And that entire group, the hominids, are more similar to one another than any of them are to a gibbon, and all of the hominoids are more similar to one another, all the apes, than any of them are to an old world monkey, and so on and so forth, in a nested hierarchy formation in both the functional and the non-functional DNA, meaning you can't appeal to common design all the way across the entire tree of life. This is not an agnostic question. The percent similarity itself is also relevant because high similarity was what was predicted by evolutionary theory, and low to no similarity was what was predicted by creationists, at least the overwhelming majority of them, because they said humans aren't apes at all. If I'm wrong on that, and there's actually a young earth creationist out there who is on the books between the 1920s and like 2005, saying that humans are going to be really similar to chimpanzees and that we're still not apes and that's common design, or that we are apes and that is common design and it's all okay, and that person isn't Todd Wood, I would be shocked, but I would love to see it. Accommodating that number now in the present and retroactively saying that it's okay, you can make a creation model that can make that work, does not erase the history.
We have a God who created. He could have created us with as few or as many differences as he chose. So whatever the answer is, that's the answer. Okay, I can take that.
This is obviously preferable to just asserting that the number isn't as high as conventional science says it is, pointing to common design and saying, okay, maybe it is really high, like 98.8% for protein coding regions, but God just designed it that way. Again, that's better, but it's still ultimately incredibly arbitrary. Like, I don't know this for a fact, but I would imagine that Rob Carter accepts that cats and lions are related, that they both descend from an ancestral sort of created cat kind. But the difference between cats and lions is larger than the difference between humans and chimpanzees, the difference between which Rob assumes are created. So how do you tell the difference between differences that exist due to divergence, common ancestry and then divergence, as in cats and lions, or created differences, as in humans and chimps, especially when the latter are more similar than the former?
The evolutionists, though, they're in a little bit of a box in that they can accept a lot of differences, but that would require them to back up the timeline, and the paleontologists won't allow the great apes in the time of dinosaurs.
Reality won't let the great apes be in the time of the dinosaurs, because there is not a single hominid fossil, not a single hominoid, not a single anthropoid, not a single haplorhine, not a single primate fossil found during the time of the dinosaurs.
So they can't back it up too much. You can't say like humans and chimpanzees split 17 million years ago, because that messes up all the paleontology.
I don't know how Rob conceives of the relationship between like paleontologists and geneticists, but those two fields inform one another. In fact, there was quite a ruckus created back in the late 1960s when two geneticists used the molecular clock in one of the first instances of this to reassess when the last common ancestor of humans and the other apes lived. And they proposed that it was 5 million years ago. They would later revise this to be a little further back in time, like 6 to 7, which is where it stands now, roughly speaking. But this really messed with the paleontologists who were at the time proposing Ramapithecus as being the last common ancestor. This was an Asian hominoid that lived 12 million years ago, so the monkey wrench was quite gargantuan. And paleontologists were not happy that geneticists were kind of bullying their way in, this is how it was perceived, bullying their way into morphology and paleontology, the keepers of the time clock. Ramapithecus ended up just being female Shivapithecus, for one, but let's not pretend that these guys won't pick fights with one another. They commonly bring evidence to light that throws a monkey wrench into the other's assessment of the data, and then it causes them to look a little bit deeper, and they end up corroborating one another quite nicely. Case in point, now, 50 years later, all of our hominins that are morphologically assessed to be hominins fit within that 7 million years ago to present range that was established by geneticists in the 1960s. Again, to the chagrin of the paleontologists, but the geneticists were right. I guess what I'm trying to say is this question only became agnostic to the conversation for creationists when they got it wrong. Anyways, Rob goes on to summarize what we've heard as far as the percent similarity between humans and chimps, and he gives all of the usual numbers. I will remind you that most of those numbers, in fact, really all of those numbers are correct. It depends on what you're comparing.
But we get to ask the question then. How different are we really? Are humans and chimpanzees 99%, 98%, 97, 96, 95%? I've heard all those numbers coming from the evolutionary community. The typical answer you have from the creationist side is 85%.
And then he says something really, really interesting to me.
How do I say this? Honestly, I've never been very comfortable with that number.
I want it on the record that I believe Rob when he says this. I think Dr. Carter is competent at what he does in the areas that he is well versed in. And I do believe that it's possible that he looked at these numbers at that 85%, 84% put forward by Tomkins, even when he was endorsing it, and that he was like, man, I really hope that this turns out to be correct because that does not sound right to me. I believe him when he says that. But I do think it's really funny that he sort of prefaced this presentation of being like, yeah, Tomkins was wrong on all of this stuff, with just a little bit of and I was never really convinced. I mean, I always kind of thought it seemed a little bit fishy. I think that's kind of funny.
I don't know if that's biologically realistic. 85%, that's a lot of difference. And if you look at chimpanzees and all the other species in the world, chimpanzees are most like us. They kind of look like us, and on some levels they kind of act like us, and they have the same food requirements and the same temperature requirements.
Man, that's pretty similar. That's about as similar as the differences between, a lion and a house cat. And those two things share common ancestors. So I wonder, hear me out. What if humans and chimpanzees are related by common ancestry? And that's why all those similarities exist, both in our physical characteristics, in our phenotype, and in our genomes. I know it's crazy, but it sure does look like the evidence is pointing in that direction. He's so close.
They are basically spiritually alive as a box of rocks. They are not intelligent. We're qualitatively very different from them.
Well, it sounds like the two big qualitative differences that you came up with are religion and intelligence. Now, religion, I can't really speak to that. I don't know if chimpanzees practice religion. People have argued that they have odd ritualistic tendencies, and they certainly behave in familiar ways regarding death. I think you can look to other animals to make a better case that we're not the only, spiritual critters on the face of the planet. Elephants bury their dead, after all, and then avoid those burial places. I think that deserves consideration. They're not completely absent on this, as you would expect if we were specially created as the only spiritual organisms on the planet. But I think intelligence is actually a much worse example. I think religion should be the one that Carter sticks to here as a qualitative difference, because humans as a species have a much larger brain case than chimpanzees, but there's not really anything that we do that is a categorical difference. We use extremely advanced tools, but chimpanzees use tools. We have advanced modified tools, but chimpanzees also modify their tools. And interestingly, that creates a prediction from the evolutionary side of things that we should see the advancement of tool types and tool complexity as we move up through the geologic column alongside hominins that are proposed to be ancestral to us. And that's exactly what we see. Plus, the vast majority of our intelligence as reflected in the types of tools that we use rely almost entirely on the cultural transmission of knowledge. An individual member of Homo sapiens from 300,000 years ago isn't dumber than any member of Homo sapiens today. They were just as intelligent as we are, and yet their tools look a lot more like the tools of the hominins that came before them than they look like the types of tools that we use today, like a computer or a car. And you can find that gradient of tools extending back through the entire lineage of hominins with virtually no breaks. It's just worth pointing out.
I would expect on first principles that humans and chimpanzees should be similar on the inside because we're so similar on the outside. So I think the creationist community says, yeah, that we should be similar. The evolution community says, yes, we should be similar.
No, though, that's not what happened. That wasn't the expectations that we saw between 1920 and 2005. That's simply not how history went down.
But no one actually knows. Before the numbers were run, no one could make a prediction. Back in the 1940s, when we started sequencing proteins, everyone expected that human proteins and chimpanzee proteins would be different because they're different species. When they started finding proteins that were the same, that was actually a challenge.
So my understanding is that humans and chimpanzees, on the basis of proteins, were more similar than anybody expected them to be. But that is not the same thing as saying both creationists and the conventional scientists expected them to be really similar. Again, the predictions were opposed to one another. Evolutionary biologists were saying that they're going to be quite similar, and creationists were saying humans aren't apes. And if I'm wrong on that, Rob, just show me a single example of a creationist from the 1940s who's saying, you know what? I bet when we sequence these genomes that humans and chimpanzees are going to be really similar. They're really similar now. The closest you're going to get is George McCready Price proposing that apes might be the degenerate descendants of humans. Eventually, Carter gets to talking about Bareminology, which is the creationist field of trying to find out what those created kinds are and how they are demarcated, either morphologically, as it used to be done, I suppose, or genetically.
Because in Genesis, God said he created things according to their kind. He didn't say how many kinds. He didn't say what the diversity within kinds was or how far apart kinds were, but we have a general idea that God created groups of organisms reproduced within themselves and not with other things. But in the field, there's no metric. There's no statistic you can say like, oh, the Cartesian Gaussian coefficient squared equals 0.32221. Therefore, these two things are two different created kinds. We don't have that number.
I would love to see the scores on that one. I'm sorry, and you know me, I don't ever rock the boat.
And that's one big point that I make here a lot on this channel, is that there is no way to consistently and in a standardized way identify created kinds without grouping humans in with the other apes. Now, there's actually no way to do it, period, because all life bleeds into all other life. But I find that to be a fun sticking point to highlight. It's one that you just saw Carter recognizes, right? There is no means by which to determine the created kinds, at least at present. But I suspect, and what Carter says next I think supports this notion, that creationists are just going to eventually give up on trying to find a standard way of identifying Baremins, and it's going to be like mostly vibes based, because they can't have humans being a part of the ape created kind or one of the ape created kinds, and they can't have too many kinds because then everything won't fit on the boat.
And so we see that some Baremins are more similar than to others. I mean, consider, all mammals are more similar to each other than any mammal is to a fish.
Yeah, and believe it or not, that's not just in their morphology or in the functional regions of their genome, it's also in the non-functional regions of their genome, which I think everybody agrees there are parts of the genome that don't do anything, even though creationists would insist that that portion of the genome is much smaller than conventional science would say. That should not exist if we're talking about a God that's doing common design instead of a nested hierarchy that's the result of common ancestry.
Why? Because God created hierarchically. And in that hierarchical scheme, some things are closer together than others. So humans and chimpanzees, they could be really close.
And that also, as I said earlier, makes it pretty tricky to tell what types of change are created types of change or change that emerged because two organisms have a common ancestry within their created kind. Like a lot of creationism with result to biodiversity has just become God did it in a way that looks identical to how evolutionary theory says it should look. What a coincidence. Actually, Carter goes on to talk about an article that he wrote, co-authored, I suppose, about how God evidently created hierarchically, just like how evolution predicted, and that complicates any investigation into Bareminology to the degree that, and maybe I'm mishearing him here, it kind of sounds like Rob is saying we should just kind of give up on finding a standardized method of identifying created kinds.
All we're saying is that if God created hierarchically, then some distinctly separate creations could be very similar. Other distinctly separate creations could be far apart. So there's no single measurement that anyone can use. You can't just say it's equal to whatever that number I quoted earlier that tells us they're two different created kinds or the same created kind.
And for my money, that acquiescence comes specifically from the fact that creationists, young Earth creationists, but I guess all of them, can't find a way to separate out humans from other apes while still maintaining a small enough number of kinds to fit on Noah's Ark.
Now, I love bareminology. I love studying the discontinuities between groups. It's really fun, really interesting. It's just a little frustrating, and that directly applies to the human chimpanzee question.
Well, yeah, I should say so. If you can't tell what the created kinds are, you can't say anything about biodiversity from a creationist perspective, and you sure as hell can't separate out humans and chimpanzees using scientific methods. You have to rely exclusively on your interpretation of the Bible, which is itself subjective.
Now, about a year and a half ago, I really stuck my foot in it. I heard about these series of videos that some group of evolutionists were making against one of my friends, Jeff Tomkins, over at the Institute for Creation Research, who's a geneticist. There's not a lot of creationist geneticists out there, and so I was worried about it. And I watched the video and said, wait a minute.
So Carter proceeds to give you the background, the story so far that I've already given to you. In his story so far, Carter articulates the reason why he thinks people were upset with Jeffrey Tomkins' methods.
And that's what these evolutionists were criticizing.
And what I'm going to say about that is that I think Carter grossly misrepresents the situation here. I think he does so because he knows the audience that he's speaking to, and also because there are time constraints on his talk. I'm going to reiterate again, the reason why people were upset with Tomkins is because his methods are, at best, incompetent, and at worst, deceitful. Like, Carter sort of alludes to this idea that people had an issue with the genomes that he picked, the fact that he was using, like, contiguous sequences that weren't humanized. No, none of that was the problem. It's best outlined by his 2015 work, but you can find it elsewhere too. But Tomkins uses separate methods that are wrong in different ways, but just so happen to all home in on this same incorrect number, which led me and many other people before and after me to think, hey, that's kind of weird. That almost seems like he used incorrect methods on purpose to home in on this specific answer that he wanted. And then in addition to that, there are weighting issues in virtually all of his papers that erroneously give this lower percent similarity between humans and chimps. All of Tomkins' methods are wrong, usually for different reasons, but in different reasons that just so happen to converge on this same theme that he is trying to underscore, that humans and chimps aren't as similar as they might have you believe. I have gone over this extensively again in this video, and also you can utilize Glenn Williamson's table or his video if you want to know in depth why Tomkins was wrong each time, and wrong in a way that is, because I just don't have a better word for it, suspicious. It's weird. Carter essentially condenses a video that I've already gone over by him, where he talks about the errors that I made in my initial analysis, which I've corrected and still haven't heard back from him on, again, and the errors that Tomkins made. He proposes again that these are like honest mistakes by Jeffrey Tomkins, which I disagree with.
That's essentially what happened. Now, it's an honest mistake. I mean...
Not really. Maybe.
Carter goes over those three numbers that he wants to see that he mentioned in the trailer, and we kind of already talked about that, how we sort of already have those numbers. We have the protein coding similarity between humans and chimps, 98.8%, compared to like the 85% for the same region for rats and mice. We have the full genome comparison, which is 96% for humans and chimps. And then there's the full raw nucleotide by nucleotide comparison, which Richard Buggs proposes hovers around 85%. But I don't think that's ever actually been done by like a team of conventional scientists actually working nucleotide by nucleotide. Carter says he wouldn't be surprised if it got up to 90% or even 95%, which I wouldn't either, but I suspect it's probably not going to be 95% because I've seen people propose that by that method, again, the human-to-human comparison is going to drop from 99.9% similar in coding base pairs to like 94%. And obviously, two humans are not going to be less similar than a human and a chimp. And then Carter proposes that we need the number for the mutational steps, the differences, like which specific mutations need to occur to explain the percent difference, whatever that may end up being for whichever number or method we're looking at between humans and chimpanzees.
Even the evolutionists can say, we can explain these differences. The creationist says, well, we don't care. great, evolutionists, we're happy for you, but that does not contradict our position here, because we don't make a prediction on this one thing. There's other things we do predict. This one we don't.
So, even if we came up with a model that step by step, gene by gene, explained how you get humans and chimpanzees from a last common ancestor, how those two genomes diverge from one another, Carter's like, it doesn't matter. We don't care about that, actually. And honestly, I think that's probably the right route to take here. If you're a young Earth creationist like Rob Carter, you might as well just say it doesn't matter. Like, we don't actually care about that, because if humans and chimpanzees are more similar than pairs that they accept, they meaning young Earth creationists accept are related, then necessarily the number of changes that have to occur in humans and chimpanzees are less than the number of changes that would have to occur between rats and mice, which creationists say are related. And they would look pretty dumb if they were like, no, you can't get that number of changes between humans and chimps. It just can't happen. Biochemically and genetically, it's not going to happen. And then they turn around and say, but it can happen for rats and mice, and it can happen for lions and tigers, and it can happen for African and Asian elephants. And in fact, it did happen, and in only 4,400 years at that. To Carter, what actually matters here is the integration time, which is the term that he uses for, insofar as I can tell, genetic regulation of the genes that are there. So Carter is taking the conventional opinion here, like this is the opinion of conventional scientists, that the primary reason why humans and chimpanzees are so different, like why we look as different as we do, is not due to the genes themselves, but how those genes are regulated.
Those things are vastly different between humans and chimpanzees.
Oh, really? How? Because my understanding is that it's actually a handful of key differences that actually make all the difference, to steal a phrase.
That's, well, something I can't even count. There's something above my head because I really don't even understand it. It's really deep, crazy kind of stuff. But if you're going to take a human, if you're going to take a human chimpanzee common ancestor and split into humans and chimpanzees, you have to have tens of thousands of genomic rearrangements. You have to move genes around. You have to change the coordination of entire genetic pathways. That is extremely difficult, and we would expect that almost all those changes are going to be detrimental to the organism.
This makes absolutely no sense to me, because Carter is accepting the conventional numbers for the differences between humans and chimps, and he's accepting that those are relatively fewer differences than pairs of organisms that he would accept are related, like rats and mice and lions and tigers, or lions and domestic house cats, or African and Asian elephants. So again, the changes that have to occur between those pairs of organisms and their common ancestor are greater than the changes that have to happen between humans and chimps as compared to our last common ancestor. And yet Carter is proposing that the latter is just unfeasible. He's incredulous about it. Whereas he is, I think, implicitly accepting the former by being a young Earth creationist who thinks that created kinds are a thing. I could ask the same question, Rob. How do you get the regulatory differences, the sequence differences, the interactions between all of the genes that are different between lions and domestic house cats that are required to explain their differences today? How do you actually get that? And it's even worse, because my question for Rob isn't how do you get that in the millions of years that it's been since lions and domestic house cats diverged from one another, but how did you get it in 4,400? Dr. Carter brings up a bunch of modern papers about how mutations can cause disease, that they're related to pathology. And this is a classic creationist argument to make. It's like, oh, but most mutations are deleterious. So how can you actually get beneficial change in organisms if change is random and most of that change is bad? It's deleterious to the organism. He sort of insinuates that there's like a mutation burden. I don't know if he's getting at genetic entropy there or what, and he sort of points out, vaguely gestures towards the waiting time problem. It's stuff that we've heard from Rob Carter before.
That's another issue for another day. I'm just putting a warning flag out there for the evolutionists that they've got some more things to account for, and they pretty much have assumed for the last 150 years that life is really simple.
The conventional literature and indeed other YouTubers who do science communication and are scientists have already covered virtually all of these points by Rob Carter, so I will direct you towards them because I'm not a geneticist, and when I do genetics, even though I eventually get there, I make mistakes along the way. So I'll again point you towards like Creation Myths and Zach Hancock, both friends of the channel. But I always find this to be a funny argument that creationists make, especially young earthers, where it's like, oh, but it's the process. Like mechanistically, how do you get these divergences when they accept the overwhelming majority of divergences that have happened in history? It's just the human chimp one that gets to them. And it's like the same natural processes that you guys accept, unless you're ICR, I guess. But like natural selection, acting upon mutation, non-selective mechanisms like drift and gene flow. You have sexual selection at play. These mechanisms work to achieve enormous amounts of change. And indeed, all of biodiversity has been supported by work both in the field and in the lab. And again, I must insist on reminding everybody that creationists accept the vast majority of this kind of change. Anyways, Carter gets into the human and chimpanzee comparisons that he's done. And it is here that he tells us that he wanted to home in on a specific chromosome because comparing the full genomes is obviously an enormous task. And so he decided to compare human chromosome 2 to chimpanzee chromosomes 12 and 13, which are sometimes called 2A and 2B, to kill two birds with one stone.
Let's look at what's called the chromosome 2 fusion hypothesis.
Chromosomes are bundles of genetic information that contain the instructions to make cells and keep them running. They're typically discussed in their unpaired numbers or in their paired numbers, and organisms all across the tree of life vary in the number of chromosomes that they have. Our superfamily is no exception. We are hominoids, so we're apes. This is the sort of category right above the great ape category. It basically just includes gibbons. Now, gibbons vary just extremely in their chromosome number. Some gibbons have 38, some gibbons have 52, while the great apes are a little bit more constrained. Great apes consist of us, the pannins, so chimps and bonobos, gorillans, and pongins. Hopefully those are self-explanatory. Humans of the great apes, the hominids, are the only species that has 46 chromosomes, while the others have 48. Okay then, so evolution makes a super simple prediction. If we humans share a common ancestor with the rest of the great apes, then one of two things must have happened. Either a fission event, which caused in several lineages of the other great apes a fission to occur, and the chromosome count to go from 46 in our common ancestor to 48 in all of them, or a fusion event occurred just in humans, where our number went from 48 to 46. Obviously, the latter hypothesis is more parsimonious because it requires fewer steps. So we have this really simple hypothesis then. Sometime in the past 7 million years, which is the approximate date that we split from chimpanzees, that our lineage, the hominins, split from the pannins, we must have experienced a fusion event. Two ancient ape chromosomes fused to form one of the human chromosomes. And since we get chromosomes from both our parents, this would reduce the total number from 48, as in the ancestral condition that still had in all of the other great apes today, to 46, the condition seen only in humans. And we should be able to find that fusion site in the human genome, like a smoking gun. When chromosome numbers were first available for humans and chimpanzees, human chromosome 2 was singled out as the chromosome that experienced this fusion event due to the similarity it had with chimpanzee chromosomes 12 and 13. This is called homology or synteny. Now, some of the best support in scientific ideas comes when you make a prediction of something before you actually investigate that thing, and then the prediction comes to fruition. This is something that young Earth creationists do not seem to understand because they constantly confuse prediction with accommodation. The prediction for human chromosome 2 showed up in 1972 in a book titled Comparative Karyotypes of Primates. Authors Cherlow and Grouchy wrote a chapter titled Karyotypes of Man and Chimpanzee, Comparison of the Topography of Bands, Possible Evolutive Mechanisms, which contains the first prediction of the human chromosome 2 fusion to my knowledge. This prediction was made just on the basis of really brute, basic homology between chromosome 2 in the humans, 2, 12, and 13, and our closest living relatives, the pannins. So what actual evidence would we have to find to support this idea? Chromosomes are structured in an orderly way. They look like X's with a centromere at the center of the X and telomeres at the ends of each X's arm. Now, these centromeres and telomeres are identifiable as such by repeated sequences of DNA that are only found in such high densities at these locations in particular. The purpose of telomeres and centromeres is also pretty well understood. Telomeres are meant to protect genetic material, and centromeres bind the chromosomes during cell division. So then, if two ancient ape chromosomes fuse to create human chromosome 2, then we should find the remains of telomeres in the middle of the chromosome, where the fusion occurred, as well as a second centromere somewhere else in the chromosome. In addition to this, the regions outside the fusion site in human chromosome 2 should share enormous similarity with chimp chromosomes 12 and 13, given these are the chromosomes proposed to have fused in the human lineage, while remaining separate in other living hominids. This pattern should also be repeatable. We should find these same characteristics diagnostic of a fusion in other animals proposed to have undergone such a process. In my previous video, I emphasized that we have indeed found the fusion site itself as well as the second cryptic centromere. You see, as I mentioned previously, centromeres and telomeres both have respective calling cards that diagnose them as such. These calling cards are repetitive sequences of DNA, and in telomeres we know it from a 6-nucleotide string, TTAGGG in forward and CCCTAA in reverse. We know this because these sequences are found in massive densities at regular old telomeres on every single chromosome in basically all animals. Centromeres are known from a more convoluted sequence that we call an alphoid repeat. So then, to reiterate with a bit more information, if a fusion occurred between two ancestral ape chromosomes, then we should find telomere calling cards in the middle of a human chromosome where the fusion occurred in forward and reverse, and alphoid repeats clustering twice in that same chromosome, once for the regular centromere and the other for the second cryptic one.
This prediction has come to fruition in glorious detail. We now know that human chromosome 2 is in fact the result of a fusion event between ancestral ape chromosomes 12 and 13, and we know this because, just as predicted, there are a bunch of telomeric repeats right in the middle of the chromosome. There is a second centromere, and there is an enormous degree of synteny, of homology, between human chromosome 2 and other non-human hominid ape chromosomes 12 and 13. I have talked about this extensively on this channel. You can see one of the many videos going through the details on that if you're interested. Yes, there was a fusion event, and this is something that a lot of young Earth creationists accept, including now, I guess, Rob Carter. In fact, just like with the percent similarity between humans and chimpanzees, Rob's kind of just always known.
I'm going to tell you something else. Just like the 85% claim that I was never really comfortable with, I've never been really comfortable with the creationist explanations for this.
Well, geez, Rob, I wish you would have said something. You would have saved me a lot of hassle. For whatever reason, young Earth creationists tend to take your word a little bit more easily than they take my word. I can't imagine why. Dr. Carter goes through the historical reasons why we suppose a fusion event occurred, and he says this.
So was there a fusion or not? Well, looking at Jeff Tomkins' work, which, you know, I really do appreciate. I appreciate this a lot.
I just think it's funny that he keeps being like, good job, Jeff, you know, for trying. Using a preprint, Carter sort of softly suggests that the vibe in the paleoanthropological community is that part of what makes us human, like in comparison to the other apes, is the human chromosome 2 fusion, which, like, yes, I would agree. That is one difference that separates humans, modern day Homo sapiens, out from the non-human apes. That is a fact. And in fact, other non-human hominins like Neanderthals and Denisovans also have this fusion, evidently. But Carter sort of pitches it in a way that's like, oh, you know, the paleoanthropologists are coming around to the idea that like all archaic humans that have this fusion are like members of Homo sapiens, which that is not at all true even a little bit.
But this paper is also hinting that all archaic humans and modern humans are just subpopulations of Homo sapiens. Ooh, very interesting. Because Neanderthals and Denisovans and all archaic humans ever sequenced have the chromosome 2 fusion and have the PAR2 on the Y chromosome. So these are distinctively human things that separate them from the apes.
Wrong!
But even if it was, I would just like to point out again that there are telomere to telomere fusion events not dissimilar from what we see in humans from a more basal ape ancestral condition. in organisms that creationists accept as related, like suids, for instance, unless he wants to propose that modern pigs and the Babirusa are members of a different kind. And I don't suspect that Carter thinks that, because he spends the next several minutes talking about the variable chromosome numbers within groups of organisms that creationists accept as related. So he talks about them specifically with regard to marsupials. And the idea from Carter's perspective is that if you have these created kinds, like kangaroos and wallabies, for example, and within these created kinds, the chromosome number varies a lot, well, then they also require chromosomal changes in the genome, like, for example, fusion events. And in fact, they require them on a much faster timescale. So Carter is sort of softly pitching, what makes humans any different? Who's to say God didn't create some kind of ancestral hominin, like Homo erectus or something like that, and then there was a fusion event somewhere along that line, just like you see in all of these other sort of putative Baremins. That's the idea. And yeah, of course that's the idea he's pitching here, because Dr. Rob Carter just spent an inordinate amount of time comparing human chromosome 2 to chimpanzee chromosomes 12 and 13. And this is the work that he did after softly accepting a much higher number than the one that Jeffrey Tomkins was presenting, because he found Tomkins's work inadequate. Of course he's going to propose that a fusion event is evident, because that's what his work is showing. So Carter downloads the most up-to-date genomes, the fully completed telomere-to-telomere human genome and the most complete chimpanzee genome, and he starts by looking for the fusion site.
And so I started searching. I searched chromosome 2 for the sequence TTA-GGG, and I found it all throughout the entire chromosome. Then I searched for CCC-TAA, the opposite, and I found it all throughout the chromosome. So I took the stuff from Tomkins' paper and I searched for TTA-GGG, TTA-GGG, and I found it in the black dots in the image in only certain places. And then I searched for a longer piece and it lit up a certain spot. And I said, that is where the fusion site is.
Yeah, so Carter just found the fusion site. Then he talks about how he runs some additional comparisons, looking at synteny between chimpanzees 12 and 13 and human chromosome 2, and he does so using BLAST, which I think is really funny because he said BLAST was a bad tool for full genomic analysis, but he learned it well enough that he found it useful for this particular comparison. So, it's funny how we end up using the skills that we accidentally acquire. And he does say this one thing that's really funny.
Extremely useful. You can do all sorts of things that the designer didn't intend for it. In fact, I've talked to him several times. He's complimented me a little bit on some of my penetrating analyses, but
it actually makes me laugh just rewatching it, because this is exactly the kind of shit that I would put in one of my videos, where it's like, oh yes, I reached out to the designer of this particular tool, to the creator of this tool, and they complimented me on some of my penetrating analyses, because it feels really good when the person who made the tool compliments you on the work you've done. And for the record, if we're talking about Ian Korf, he's also said some very nice things about my analyses, Rob. So you can check out the video, Rob's interview, if you want to see him go step by step through what he did. I think it's really interesting personally, because obviously I'm invested in this particular conversation, but I'm going to leave you with one of his biggest punchlines, because as I said, after he finds the fusion site, he goes and he compares the full chimpanzee chromosome 12 and 13 to human chromosome 2. He's looking for overall synteny, right? So if a fusion event did occur, then it's not just that human chromosome 2 should have that fusion site in the middle and those two cryptic centromeres. Human chromosome 2 should just be really similar to the two fused ancestral ape chromosomes, which are maintained in modern day non-human hominids. So Rob does that search, and after troubleshooting and figuring out how to best run this analysis, he finally gets his results and he hits you with it like a drunk truck driver.
What do you think you're going to see? That. Oh, yes. Chimpanzee chromosome 12 and 13, or 2A and 2B, align beautifully on human chromosome 2. Oh, and notice that the start of chimpanzee chromosome, this one here, the chromosome 12 is backwards, chromosome 13 is forwards.
He actually goes on for a long while explaining basically the support that there was a fusion. And he does this without outright saying, like, guys, there was definitely a fusion. He instead ends the presentation by saying,
in the creationist community, we need to have a discussion. Was there a fusion? And when did it happen?
I think Robert Carter's work and interview here speaks for itself. I think that he thinks that it did happen, but to be fair, he doesn't come right out and say that, not bluntly at least. But he does say that he thinks if there was a fusion, that it would have had to have happened on Noah's Ark, which I think is a fun, world-building bit. And then he goes on to say he does actually want to continue work on the overall percent similarity between humans and chimpanzees. Why might he want to know that, you might be thinking, if it supposedly has no bearing on this question of creationism and evolutionary theory?
I don't know, just as a nerd, I want to know that number.
Rob he cracks me up with this. He's one of my favorite creationists for this reason. He's just so fun. I think he's a fun science communicator. And that's what he's doing. He's right here communicating some science. Is he a young Earth creationist? Oh, you bet your sweet bippy he is. But the science that he presented here, at least methodologically speaking, the data themselves and his analysis, are pretty good. Obviously ends the presentation by talking about how he is a creationist and how one of the reasons why he thinks the way that he does is because he can kind of appreciate the complexity of life, which is something that he thinks that evolutionary biologists don't do, or that conventional science doesn't do.
Evolutionists, and I'm making them mad just by saying this, but they have downplayed the complexity of life from day one. Always, life has been simple, and the more complex life becomes, the harder it is to explain.
Oh, dear, why I oughta. He's making me so mad with that. I think he's wrong. I don't think that conventional science has downplayed the complexity of life at all. And I think all you have to do to see that is take a little deep dive into the literature of whichever field you're curious about. There's so much fighting about every little myopic topic because we as scientists want to know the truth, and everybody has ideas that they think work a little bit better. They have their pet hypotheses. Of course, this doesn't change the basis of the science, but we all have hypotheses about new areas that we're researching, such as the nature of the beast. I also happen to think that science is dead the moment you encounter a tough question and your answer is, well, I guess God just must have done it that way. So Rob kind of pitches you his textbooks and his DVDs briefly, and he said he has this one cute little line at the end where he's like, and if you ask the secretary in your purchase order, I will autograph a copy for you, which I think that's really fun. And if I were to ever order that textbook to review myself, I would ask Rob Carter to sign it. So if you're out there and you're thinking about it, I think it would make his day, and I think that you should ask to have Carter sign your copy of the book.
In fact, if you order it and you put a little note in the sale thing, the secretary might bring me one to sign for you. I've signed a lot of them already. Just getting that out there for you.
But then he ends this presentation with something that I find very, very telling and very, very interesting, just like I find all of this very interesting.
So, there you have it. Reassessing human-chimpanzee genetic similarity. The answer is not easy to come by. There's a lot of information out there. It's really hard stuff, but honestly, it also doesn't matter because I can take it either way. That doesn't mean my God is not the creator. Okay, that's it. I hope you enjoyed that.
Who is saying that? Who is saying that humans being 98.8% similar in the functional regions of the genome to chimpanzees means that you can't be a Christian and believe in the Christian God? Who is out here saying that you can't accept human evolution and the ancient age of the earth and all of conventional science and not be a Christian? I know who's saying it. It's the young Earth creationists. This is a common theistic evolutionist W. I work in a department of biological anthropology. I work with people who study human evolution specifically. And there are plenty of religious people who I work with. There are Christians, there are Jewish people, there are Buddhists. Here on this channel, I am a firm supporter of the theistic evolutionists. I'm a firm believer that you can accept all of conventional science and still be a Christian, or that you can be a Christian and support all of conventional science. I just think that that is something that is totally fine. You're never going to hear from me that the similarity between humans and chimpanzees chimpanzees and the reality of humans as apes who descended from other apes means you can't be religious. Now, Carter does end with a Q&A, and there are some friendly, familiar faces in the Q&A that I would love to go over here, but I really don't think that now is the time. There are some better places that I can think to bring those questions out. So I'll just leave you with this. I am so unbelievably happy that a silly little YouTube video of mine going over a couple of experiments that I ran has had such a large impact on the creationist argument landscape. Again, if I hadn't had made that video, then Carter wouldn't have done a deep dive and concurred that Jeffrey Tomkins was wrong in all of his analyses from 2011 onward on both the human-chimpanzee genomic similarity and the human chromosome 2 fusion. And now that creationists are hearing it from not just secular sources, but also other young Earth creationist sources, I'm starting to see Jeffrey Tomkins' number disappear. And I'm glad to see that, simply because Jeffrey Tomkins' methods and his results were wrong. And I don't like seeing wrong information spread recklessly across the internet. God knows we get enough of that already. It delights me to see Dr. Robert Carter, a young Earth creationist, be so transparent and so honest with his analyses. And it makes me even happier to see him writing those up and doing interviews about them and trying to correct the record a little bit. I am so happy that the future of creationist genetics seems to be more in Robert Carter's hands than it is in Nathaniel Jeanson or Jeffrey Tomkins' hands. Because with Rob at the helm, it's going to be more correct. And as someone who loves conventional science and who loves genetics and biology and human evolution, I'm going to take what I can get. And of course, I'm empowered, because this means minds can be changed. Thank you so much for being here, my gentle and of course very modern apes. If you like what I do here on this channel, and if you stayed till the end for 2 hours, another 2-hour video from me, please consider supporting me in the free way by liking, commenting, and subscribing. It actually does help the channel. It helps push me into the algorithm of other people, and it's free, so please do it. If you want to support me in a more financial way, you can, of course, join the Patreon. Those guys get early access to videos once in a blue moon, but they always get their names at the end of videos. And so, from the bottom of my gentle and, of course, very modern heart, I want to thank you for being such a curious primate, and I'll see you real soon next time.